everybody. Thanks for joining in today's webinar, which is about uncovering protein function with UniProt. So yeah, my name is Michelle McGrain, and I'm the curation coordinator with UniProt at EMBL-EBI. And so the talk is divided mainly into two sections. So in the first part of the talk, I will describe how the information that we collect is added to the UniProt resource. And in the second part of the talk, then I'll show you how the information is organized on the website and how you can access functional information. So what is UniProt? So UniProt is a resource of protein sequence and functional information, and it aims to provide comprehensive and high quality data. And all of the data is made freely available on the UniProt website at uniprot.org, and the data is released every 8 weeks. And the UniProt resource is produced by the UniProt Consortium, which consists of groups at EMBL-EBI in Kingston in the UK, at the Swiss Institute of Bioinformatics in Geneva, and the Protein Information Resource at Georgetown University and University of Delaware in the US. And so the three groups together work together to contribute to producing the UniProt. So why do researchers use the UniProt resource? Well, we know that there are huge amounts of data being produced, and understanding the function of proteins is crucial for a lot of research. But it can be difficult to keep up with the amount of data that is being produced. And this data is often collected in disparate sources. So there's information in the literature and also spread across different resources. So UniProt tries to act as a central resource to collect protein information. So it aims to provide a collection of high quality and comprehensive data. And it also aids scientific discovery by collecting all of this information and organizing and interpreting the data and making it easily accessible for users. And this saves researchers hours of work in having to collect all of this information from multiple sources for themselves. And UniProt also provides a range of tools for protein sequence analysis, including things like blast searching and tools to allow for alignment of protein sequences. And as well as all of the information that is collected into the UniProt resource, UniProt also provides links to more than 180 other resources, meaning that UniProt can act as a central hub so that you can come in and look at the information that's stored in UniProt, but also use it as a starting point to then explore related information in other resources. So UniProt is a resource of protein sequence and function, and the sequence data are organized into a number of different resources. So the UniProt archive, or UniPark, is a non-redundant archive of publicly available protein sequences collected from across different databases. The UniProt Consortium also produces the UniRef, which are UniReference clusters, UniRefs. So these cluster protein sequences at different levels of identity. For proteins that are part of completely sequenced proteomes, there's access provided to them through the Proteomes portal. And the centerpiece of the activities of the consortium is production of the UniProt knowledge base, and that's what I will concentrate the rest of the talk on. So there are two data sets within the UniProt knowledge base, or UniProt KB. So there's the reviewed SwissProt section and the unreviewed Tremble section. So in the reviewed SwissProt section, all of the data are manually reviewed, and the data comes mainly from scientific literature, and that's coupled with manually reviewed results from computational sequence analysis. And then the unreviewed Tremble section, there's no manual review of the data. So the protein sequences are imported from various sequence repositories, and then there is additional data which is generated from computational pipelines. So where do the sequences come from? So most of the sequences that are in the UniProt resource come from translations of coding regions in nucleotide sequences that have been submitted to the databases of the INSDC, which is the International Nucleotide Sequence Database Collaboration. And that includes ENA at EMBL-EBI, GenBank at NCBI, and the DNA Databank of Japan. And so UniProt imports those coding regions into the database. In addition, we also import sequences from the Ensemble and RefSeq resources. So there are many genomes that are submitted that don't have any coding regions annotated on them at the point of submission. And resources like Ensemble and RefSeq run their own gene prediction pipelines and attach this information to the genomes, which means that we can then import these proteins into the resource. We also import protein sequences from the protein data bank so that we can make sure that for every structure that's in PDB, there's a corresponding protein record in UniProt. And we do also accept submissions of directly sequenced proteins. So this is a very tiny proportion of the sequences that are found in UniProt. So these are, but they're generally very interesting sequences because they're often from labs who are really characterizing these proteins as well. So they're often sources of novel data. Very often researchers will submit to us pre-publication so that they can have an accession number to use in their publications, and we can hold the data confidential until their papers have been made public, and then we will release the data and curate the sequences with the information that they have published in their papers. And so all of these sources together are used to populate the UniProt database, and sequences go into the unreviewed Tremble section, and then there's a subset of those sequences will be manually reviewed and added to the reviewed SwissProt section of the UniProt resource. So I'll just say a little bit about what this manual curation process involves. So UniProt has a team of curators who are responsible for the information content of the database. And so there's a lot of effort goes into curating the sequences. So for merging sequence reports from the same gene and the same organism into the reviewed entries. We also attempt to match the genome so that the sequences are as accurate as possible. And so this means that curators will often correct sequences if they believe that they're incorrect. And all of the discrepancies between multiple sequence reports for a given protein are documented so that it's clear if there are differences, what the reasons for those differences are. So there's a lot of work goes, like as I say, goes into maintaining the sequences, but then that ensures that any further sequence analysis that's done is as accurate as possible. So the curators also run the sequences through various sequence analysis programs to predict features like post-translational modifications in a protein, any transmembrane domains, topology, and to identify other important domains. And all of the predictions are manually reviewed, and only relevant results that makes biological sense will be selected for inclusion into a record. And then these predicted features are combined with experimental data to provide a complete overview of the sequence features of a protein. And most of the data that is added by the curators is coming from the scientific literature. So during the literature curation process, the published results which are found in the scientific literature are extracted and converted into a format which can be easily understood by users, but is also computationally tractable through using controlled vocabularies and ontologies to represent the data. And the data from a single paper is then combined with data from other papers and from other sources to provide a complete overview of information. And any conflicting or erroneous data can be highlighted and corrected. And this process then enables us to provide a complete overview of a particular research area. So during literature curation, the experimental findings will be critically evaluated. And as I said, it's all compiled into a comprehensive report and represented using controlled vocabularies where possible. And all of the information is linked back to the original publication so that it's clear where the information, the original source of the information is. So how do we find literature? So we do monitor new publications that come out in a set of high impact journals. We also make use of an additional bibliography. These are papers that we haven't actually curated ourselves, but which we have imported from other resources. We also make use of literature databases. We have a very active user community who also provide information about new papers that may be of interest for curation. And we also have a facility, which I'll say a little bit about later, to allow for community submitted literature. And we also make use of text mining tools to identify relevant papers. So we make use of the PubTator tool, and we've been using this primarily for our weekly literature triage. So we use PubTator and we run it over a set of, like as I said, these high impact journals to help us to identify relevant papers. And we've also been using LitSuggest. So both of these are freely available tools from the NCBI. And we've been using LitSuggest quite successfully to identify domain-specific literature, so literature that's specific to a certain area. So for things like, we've been using it for things like identifying novel variants in the literature, also novel enzyme reactions. And we've been using those two tools quite successfully. And I've given the URLs there if anybody is interested in exploring them a bit further. So once curators have identified literature, then there's a whole host of information that's collected from the papers. So from things like protein and gene names through to the functions of proteins for enzymes, what activities they, what any reactions that they catalyze, any cofactors that are required for those reactions, where in the cell a protein is expressed, what tissues it's expressed in, if it interacts with any other proteins. And then at the level of the sequence, there's things like, are there other splice variants produced, and any domains, regions, or important sites in the sequence are also collected. Important sites that may be targets of post-translational modifications, and important residues for catalysis are also identified. And curators take all of this information and they add value to it by collecting it together and organizing it and making it easily accessible for users. So manual curation is very important in ensuring that we have a body of high quality data. But so this slide shows the growth of the Tremble resource over the last number of years. So it's clear that manual curation can't keep up with the speed at which new sequences are being produced. And even if it could, many of these sequences have never been experimentally characterized. So there is no experimental data attached to them. So it's important that we have ways of providing some information about the huge body of uncharacterized proteins that are in the unreviewed Tremble section. And so to do this, we have developed a number of automated methods. At the level of the protein sequence, there are a number of automated sequence analysis programs run to predict signal sequences, transmembrane domains, coils, and intrinsically disordered regions. And we also make use of the Interpro database and the member databases, ProCite, SMART, and PFAM, to predict automatically domains, important domains on the protein sequence. And then we also have two automatic annotation systems which are used to add information to unreviewed records. So the first of these is called the UniRule system. And in UniRule, although the system itself is applied automatically, the rules are manually created by curators. And in contrast, the ARBA system is completely automatic. So that's based on association, rule mining, and selection techniques. And so those two systems together are used to add information to the Tremble records. And recently we've been exploring other methods that can be used to improve the information in the unreviewed records. And so an example of this is a collaboration that we've had with Google Research to develop Prot LLM, which is a natural language processing model. And it's been used to try and predict names for proteins. So it's been applied to uncharacterized proteins, which previously didn't have a protein name. So in the database, they would have been called uncharacterized proteins. And so the slide just shows an example of such an entry, which previously would have been called an uncharacterized protein, but now has a name predicted based on this Prot NLM system. And together, all of the automatic annotation systems provide information for almost 90% of the unreviewed records in Tremble. And as well as all of the information that's added directly by UniProt, we also make use of large, publicly available large-scale data sets, which are available in other resources. So we also incorporate variation data from a number of variation databases, and that complements the variants that are collected by the UniProt curators from the literature. And the same is true for mutagenesis, which is collected both from the literature and also imported from other resources. And we also have proteomics identifications from publicly available mass spec databases, and also post-translational modifications and AlphaFold predictions. So it's clear that the information in UniProt is coming from multiple resources. So it comes from researchers, from scientific literature, from various computational pipelines, from other databases, from just knowledge that curators have. Sometimes we combine information from multiple sources. And so it's really important that we have a way of attributing all of the information back to its original source. And so to do that, we use the evidence and conclusion ontology. So that this allows us to link all of the information back to its original source. And so, for example, if a paper shows that a protein is acting as a tyrosine kinase, then using the appropriate eco term, we can link that information back to the original publication, and also provide information that there's experimental support for that function, and that it has been manually reviewed. So just to summarize the first part of the talk, the UniProt KB is divided into two sections. We have the reviewed SwissProt section, which has just over 500,000 records. And in that section, the information comes primarily from the scientific literature and from sequence analysis tools. And all of the data in the SwissProt section has been manually reviewed. The unreviewed Tremble section is a much larger section with over 250 million records. And while there is no manual review, there is information added through various computational pipelines. And for both sections, all of the data is linked back to its original source so that it's clear where the information has come from. And this allows users to see whether information has been experimentally characterized, or if it has been predicted, and if it's come from a prediction, which program it has been used, or if it's experimentally characterized, and which paper is the source of the information. So I'm just going to move on to the second part of the talk. And so now that we've gone through where the information comes from and how it's added to the resource, I'm going to show you where you can find some of this information on the website. So this shows the homepage of the UniProt website at uniprot.org, and there's a search box on the homepage. You can do simple queries. It also supports advanced queries for users who want to do more complex searches. So this is the search box. And so it will allow you to search with anything you want, a gene name, an accession number, if you have a term of interest to search. And once you've found your protein of interest, you will be taken to the entry page for that protein. And there's a menu across the top of each entry page, which allows you to choose how to view the entry. So you can jump to particular sections, and I'll go through some of those in a bit more detail later. And on the left-hand side of each entry page, there are quick access tabs so that you can quickly jump to specific sections of the entry that you're interested in. So for example, if you're interested in the function section, then you can click on the function link and it will take you to the function section. And in the function section, there's some free text information about the function of the protein that's been compiled from multiple papers. Like I say, everything is linked back to its source publication. So there's links to the source publications for the information. And this particular protein is an enzyme. So there is the reaction details are provided. We use the RIA reaction database for that. Within a reaction, you can click on a reaction participant to access further information. So the reactants are linked to EBI, which is a database of small molecules. So you can click through to the EBI database to find more information about each of the reactants. And as I said, everything is linked back to the original publication, and there's just a summary button beside each piece of information, which tells you how many publications support each piece of information. And you can click on that then to access the full publication details. And it will open out a box which lists all of the papers so that you can see which papers have been used as the source for the information. And there's also links to literature databases and also links to other entries where that paper has also been used. And as well as the free text information, there's also gene ontology terms provided. So if anyone is not familiar with the gene ontology, this is a controlled vocabulary to describe the molecular functions, biological processes that a protein is involved in. And there's also a cellular component part which describes where the protein is found within a cell. So you get we use this Go ribbon, which provides a nice high-level overview of the terms that have been assigned to the entry. So you can quickly get an overview of the various functions and processes which the protein is involved in and where it's found in the cell. And in addition, then all of the individual gene ontology terms are also provided. And there's information about the source of each term. So UniProt is a member of the Gene Ontology Consortium, along with many other databases. And we exchange information with other consortium partners. So the gene ontology terms that are provided in the UniProt record are a combination of terms that have been assigned by UniProt curators, but also terms that have been assigned by other databases and that have been imported. And that means that there's no duplication of effort, of curation effort across the different resources, but we still provide a complete picture of the terms that have been assigned to a particular protein. If you're interested in knowing where the protein is active within a cell, you can go to the subcellular location section, and this provides the list of subcellular locations where the protein has been identified. And we're using Swiss Biopics so this provides it's a graphical tool which has been developed at the Swiss Institute of Bioinformatics, and it's a freely available tool. It's an interactive tool which allows, and it provides a number of different cell types across the whole taxonomic space. And I've given the link to the Swiss Biopics website there if anyone is interested in having a look at it. So when you're on this section, you can hover over a location and it will highlight it on the picture so that you can see exactly where it is. And we also provide information about where protein is expressed within what tissues it's expressed in. There's some information captured from the literature, and there are also links to more specialized expression resources for more detailed information. And so you can click on that section and then you can be taken to a particular expression database. In this case, this is the Human Protein Atlas. So you can see that this protein, it gives a list of which tissues are expressing the RNA and the protein. So for some extra information. There's also information provided about whether a protein is involved in a particular disease. So this so that you can find information about diseases that are associated with genetic variations in a protein. So in this particular case, this protein is involved in a disorder called Charcot-Marie-Tooth disease, which is a disorder of the peripheral nervous system. So there's a free text description of the disease, and also there's information about any variants within the protein sequence which have been associated with the disease, which have been captured by the UniProt curators from the literature. And to be able to explore genetic variation of a protein more fully, there is also a variant viewer provided. And this shows all of the variants that have been captured by the UniProt curators from the literature, but also combined with variants that have been imported from a range of variation databases. So they're shown in a graphical view, and there's a filter on the left-hand side of the page, which will show you, it allows you to filter both by consequence and also by provenance, if you want to filter for particular sources of data. And then there's also a table which gives a more full description of the variants. And if you're interested in a particular variant, you can click on it in the graphical view, and it will be highlighted in the table underneath. And so I just wanted to show you as well, I mentioned before that we have automatic annotation. So this is an example of an unreviewed Tremble record. So when the Tremble records come in, there's very little information apart from the sequence and some information that's been provided by the submitters of the nucleotide entry. But when the automatic annotation is run over the entries, then it can provide more information. So in this case, there's a reaction which has been provided by automatic annotation, and it's come from the ARBA system. And so if you're interested in exploring information in other resources that's relevant to your protein of interest, then you can click on the external links tab in this bar, which is found on every entry page. And so this gives you easy access to information in a range of other resources. So UniProt links to more than 180 resources, as I already mentioned. So and the resources on the web page are divided into specific categories to make them easy to find, so that you can start on UniProt and then jump out to other resources and find extra information. So if you're interested in exploring domains or sites in the protein sequence, you can click on the Family and Domains link in this on the left-hand side of the page. So this will allow you to view any domains, regions, and sites in the protein sequence. And you can also filter, so that if you only want to see a particular subset of these, you can filter out the ones that you're not interested in. If you're interested in information to do with any important post-translational modifications of the protein or processing of the protein, there's the PTM and processing section. And so I've shown you already how we attribute data that's come from a publication. So in this case, there's a phosphorylation site in this protein. So it hasn't been shown directly for this particular organism. So the tag is by similarity. And if you click on that, you can see that it has been inferred from sequence similarity, and it will take you to the original source of that post-translational modification. So again, making it easy to trace the information back to its original source. And if you want to see, we also provide a feature viewer, which is highlighted here again in this bar, which is found on the top of each entry page. And this is a graphical viewer for sequence features. So on the left-hand side, the features are divided into categories, and then if you are interested in a particular category, you can expand the category to see the more specific data. And this tool allows you to view all of the important sequence features in the context of the other features in the entry. So it can be a very powerful tool. So for example, in this case, here there's an active site, so this which is important for catalysis, which is that residue 417. And you can also, within the feature viewer, you can also view the variants. So you can see that there's also a variant occurring at the same position. And so you can start to see why a variant might affect a protein if it's altering an active site, that's very likely going to have consequences for the function of the protein. And the feature viewer also includes an interactive 3D structure viewer, and so that you can view important features within the context of the folded protein. And also AlphaFold predictions are included for those proteins which don't actually have a solid structure themselves. And I'm just going to cover very quickly what papers, how you can explore what papers the information in an entry comes from. So there's also a publications tab, which you can use to explore the literature for a protein. And if you click on that, you can see that there you get a list of the references. And on the left-hand side, you can see the papers that have been used as sources of information. And there's also a list of papers which are classed as computationally mapped. And these are papers that have not been curated by the UniProt curators, but they've been imported from other resources. So this allows you to more fully explore the literature for a particular protein. So for reviewed papers, these are papers that have been curated by UniProt. There's information provided about what has been cited from each paper. And you can filter the papers based on the category of information that they provide and also on the study type. So they're classified into small and large scale studies. So if you want to just look at the small scale studies, for example, you could filter out all of the large scale papers. For papers that have been automatically imported, they are also categorized and the source of each paper is provided so that we attribute these back to the original database. So for example, in this case here, this is a paper that has come from the Intact database. So although it hasn't been curated by UniProt, we can provide it and show that it has some interaction data relevant to this protein. And if you do notice that a paper is missing and you would like to submit it, you can use the Add a Publication link, which is on the entry page of every protein. And if you click on that, you'll be taken to a submission page, and which will guide you through the submission process. So you just need to provide the accession number of the entry that the paper should be associated with and to sign in with an ORCID ID. And using ORCIDs allows us to authenticate the submission and also allows us to give credit to whoever has provided this information. And so all of these papers that are submitted by the community then go on to appear in the publication section. So there's a separate section for those. So the papers will be added and the ORCID ID of the submitter is linked to the submitted paper as well. So just to summarize, so UniProt provides a wide range of functional information. And the information comes from multiple sources, including scientific literature, sequence analysis tools, and various automatic annotation methods, and information that's been imported from other databases. And all of the information is linked back to its original source, and everything is freely available from the UniProt website at uniprot.org. And for accessing the data, so as I mentioned before, the UniProt is released every 8 weeks, and all of the data is provided via the website. The data is provided in a range of different formats, and the website supports both simple and complex queries. And it's also possible to programmatically access the data. And I've given the link on the slide if anyone is interested in that. There's lots of details and lots of examples given to help with programmatic access. If you're interested in keeping up with what's happening in UniProt, we have a blog, and you can also follow us on various social media. And if you need any further help, then there is a help link on every page. And also there's also a feedback link if you want to provide any feedback about the website. And if you click on the help link, you'll be taken to our help center. So there's lots of documentation there. And if you can't find what you're looking for, then please do get in touch. And we have a very active help desk, and we'll be very happy to help if you're having any problems with accessing the data. And finally, I'd just like to acknowledge the UniProt team. So the UniProt is a big project. There's lots of people who contribute to the resource. So I just like to acknowledge all of them, and also to acknowledge our funders who make all of our work possible. And I did very briefly touch on automatic annotation in UniProt. And if anybody is interested in learning more about that, we do have a webinar coming up in January about automated annotation in UniProt. So if you're interested in hearing more about that, then please do register for that webinar, which will go into the automatic annotation in a lot more detail than I've done today. And yeah, so that's it. Thank you everybody for joining in today and listening. And if anybody has any questions, yeah, I'll be happy to answer them.
Thank you, Michelle, for the webinar today, and thanks everyone for joining in and sending your questions as well. So we have quite a few questions in there. I can start reading them out. So how does UniProt collect the data, and what kind of algorithms are used for structures and sequence alignments?
Yes, well, I think probably I've answered that question already during the talk, but for the manually curated data, yeah, it's coming primarily from scientific literature. For the automatic, so when we do the automatic annotation in Tremble for the sequence analysis, we're using TM-HMM and Phobius for predicting transmembrane domains. We use the COILS program for coiled coils, and for intrinsically disordered regions, we use MOBI DB Lite. And yeah, so the manually curated information comes primarily from scientific literature and sequence analysis tools, and the automatic annotation is coming from our, these pipelines that predict domains and various features on the sequence, and also through our in-house developed automatic annotation systems, which are UniRule and ARVA, and also this ProtNLM system, which we've been using so far for predicting protein names.
So which company did design the website for UniProt?
Well, we have, UniProt has both curators and developers, and the developers, we have web developers, so it's been developed by the web team within the UniProt group.
Great, thank you. So next one is, thank you for the nice talk. Do you know when the ProtNLM collaboration with Google Research will be released? Do you have any other source of prediction for uncharacterized protein that UniProt uses?
So the ProtNLM data is already public. So that is already there. Obviously, it's being improved all the time. So it was first released earlier this year, and then it's gone through several iterations to improve the data. And yeah, I didn't put it into the slide actually, but there is a preprint as well to describe the method. So I can pop that in the chat, or we can send it out later on. But it is on the UniProt website, so you can go in and have a look for it as well. But there is a preprint if anyone is particularly interested in the methodology. Yeah, that is also available.
Thank you. How can we contribute to UniProt KB as a volunteer?
Well, I did mention this community submitted publications. So you can, if there is any, if you notice any missing publications or information, you can click on that link, and as long as you have an ORCID ID, you can go in and submit the publications. And also there's the facility to provide some information about what the publication is, what kind of information the publication is providing. And that is then the curators will then use that as a source as well of possible papers to curate in the future.
Are PTMs shown in UniProt are experimentally proven or predicted computationally?
It's a mixture. So we do extract experimentally derived PTMs from the literature, and then we do also use prediction programs to predict some PTMs, so things like end glycosylation. We do have a prediction program for that. So it is a mixture, but the PTMs will be linked back to the original source, so it's always clear whether something is coming from the literature or if it's a prediction.
How best to analyze genomic coordinates? Is it available in UniProt?
Yeah, so actually that is something that has very recently been added. So when you go into a particular entry, when I showed this bar that goes across the top of the page, there is a new addition to that called genomic coordinates. So if you click on that, you can go in and you can access the genomic coordinates directly in UniProt.
Thanks. Is GeneBank ID and UniProt are distinct from each other, or they are related somehow?
So they are different. So GenBank is nucleotide sequence resource, which is maintained at NCBI. So they have their own set of identifiers. If we import sequences from there, we do have a cross-reference back to the original nucleotide record. So the entry will have its own UniProt identifier, but it will also, within the body of the entry, have a link back to the original GenBank record. So it will have the accession number in the entry as well. So they're two different sets of identifiers, but we do link between each other.
Would it be possible to make the peptide search with less than 7 amino acids, which is the current setting?
Honestly, I don't know. Again, I can find that out if that is something that somebody's interested in. Yeah, so I don't know what the technical challenges are for going below that threshold. And I'm not aware of any plans at the moment to shorten the threshold. Yeah.
Right. So next one is, when looking for the specific context of a protein in an associated publication, often I cannot find the protein mentioned in the linked publications at all. Do the automatic annotation algorithm find these proteins specifically, or do they have looser terms for identifying a related protein instead?
So I guess this is referring to the linked publications that we have in the computationally mapped section. So they are so for the papers that are manually curated by UniProt curators, then we have obviously looked through all of those and confirmed that the paper is really dealing with the protein that it's describing. For the papers that are in the computationally mapped section, those papers have come from other resources which have curated them and linked them to the relevant proteins. So they have gone through some kind of QA there at the other resources. So either they've been curated by UniProt or they're coming from another resource where they've already been linked to that particular protein.
Okay, so next one is, can a subset of protein annotations can be made from the whole database of proteins in UniProt for a given protein sequence? Do you have tools in UniProt for this?
I mean, so I'm not sure if this answers the question, but I mean, it's possible to do advanced queries on the website and to pull out particular subsets of data. So if you're only interested in certain kinds of data, it's very easy to drill down and get that kind of data. And if whoever asked the question, it needs some help with accessing particular subsets of data, then you can always contact us at the help desk and we'd be happy to help with specific queries.
In UniProt, is it possible to convert accession number to get UniProt IDs?
Yeah, so we do provide an ID mapping service, and so if you have a set of identifiers that are not UniProt identifiers that you want to convert to UniProt identifiers, you can use the ID mapping service and just paste in your set of identifiers and it will return the corresponding list of UniProt identifiers.
For a given genome, can we shortlist all the proteins with experimentally validated protein function?
Yeah, so again, that is something that would be possible to do as an advanced query on the UniProt website. So you can start off by querying for your species of interest, and then you can query within that species for only entries where the function has been experimentally characterized. So that is possible to do. And again, if you're having problems trying to find that kind of data, if you contact the help desk, we can provide you with a query. But it is quite easy to do on the advanced search on the website.
So why is there only one AA for the catalytic site?
I don't know if this is, I'm not sure if this is referring to the example I showed where there was the unreviewed record where there was a catalytic activity predicted from automatic annotation. And so the way the automatic annotation works is it makes use of the reviewed entries in the SwissProt section, and it also makes use of InterPro, which is a database of protein families and domains. So we can use InterPro to group proteins into particular families, and we can then look and see what kind of information entries in that family share in the reviewed SwissProt section, and then extract that information and apply it to records in the unreviewed Tremble section, which are also part of that group. And so in that case, for the example I showed, that entry obviously belonged to a protein group or family where the reviewed SwissProt records already had that catalytic activity shown. And so because that horse entry belonged to the same group, it was possible then to predict that it would also have the same catalytic activity. So I hope that answers the question.